Okay, so now let's talk some more about how gene expression gets regulated in eukaryotes. And one of the biggest takeaways from this for you is just to realize that there are a lot of different points throughout the process of transcription and translation where eukaryotes very finely control gene expression. So we're going to take a look at a couple of examples specifically just to give you a good starting tool set for understanding what might be happening in specific contexts where gene expression is getting regulated. So, this again, if you think about all the cells in your body, all the cells in your body have the exact same genetic information in them, but those cells all express different genes, right? We have different tissues, different organs, and most genes are differentially expressed across those different tissues and organs. So, basically, on the left here, I've got a summary figure that shows you all the different steps at which gene expression can be regulated. And all of these points can be regulated to control which genes get expressed where and when. So, for example, one big area for regulation of gene expression happens actually at the level of the chromatin. So we'll look at that in greater detail. Along with that, we'll look at how transcription initiation is more finely regulated in eukaryotes, and then we'll also examine some mechanisms of post-transcriptional regulation, so further down in this process. So there are two major types of modifications made to chromatin that you need to be aware of, and the two major modifications are referred to as histone acetylation and DNA methylation. And so I'm just going to give you a very brief overview of how these work and their impacts on transcription rates. So hopefully you remember by now that histones are the proteins that form some of the organizational structure for DNA and for chromosomes. And histone proteins have a set of tails that protrude out from the DNA that's wrapped around them. And when those histone tails have acetyl groups added to them, that actually repels histone groups from each other and exposes more of the DNA that's between them and facilitates gene transcription. So acetylation is associated with an increase in gene transcription rates. And then in contrast, another thing can happen to modify DNA, which is that methyl groups can be added to the sugar phosphate backbone of DNA. And when those methyl groups are added, that's an extra molecular component that will block binding by RNA polymerase. So DNA methylation is associated with a decrease in gene expression or gene expression inhibition. So one place where this is observed is in females. Actually, over the course of development, females biologically have two X chromosomes. But female cells don't actually express both of those X chromosomes. So during development, one of the two X chromosomes in every cell winds up being inactivated, and it's inactivated through methylation. And again, as I talked about previously, some of these forms of activation and deactivation are actually inherited from parent to offspring. So they are examples of what's called epigenetic inheritance. So epi meaning outside of genetic. So these chromatin modifications can be passed on, which is really a profound shift in our thinking away from thinking about inheritance happening strictly through DNA. Turns out it doesn't. So that's modifications to DNA. Here's some examples of how the initiation of transcription gets regulated through the presence of transcription factors and enhancer regions. So here's just a reminder of the process of transcription in eukaryotic organisms, right? So we've got much more complex genes, and the DNA sequence itself will consist of a series of exons and introns that then get spliced, further processed, capped and tailed, and then sent to the cytoplasm for translation. So I talked previously about the general transcriptions that bind to the core promoter region and facilitate binding by RNA polymerase III. And there's basically 6 major types of these general transcription factors. However, there's also thousands of specific types of transcription factors, and these will also bind to other regions of DNA that are associated with any particular specific gene. So this illustration here that I showed you earlier shows an additional set of activator proteins that are binding to other regions of DNA called enhancer regions, and all these pieces have to come together in a precise organization to facilitate binding of RNA polymerase and facilitate transcription. And these factors can behave very similarly to what we saw for repressors in prokaryotic organisms. Some of the transcription factors will function as these activators. Other transcription factors will act as repressors. And so really it's the specific combination of activators and repressors that are present in any particular cell that determine which genes are turned on within that cell. So if we're thinking about this, we'll just look at an example comparing a liver cell to an eye cell. And if we look at the gene regions for each of these cells, they're going to have associated enhancer regions as well. And the enhancer regions are going to be specific to each gene. So one of the major protein types that gets produced in the liver is called albumin. And one of the major protein types produced in the eye is called crystalline. So the albumin and crystalline genes each have distinct enhancer regions. So if we were to look in a liver cell nucleus, what we would find is that the appropriate activators are present to facilitate transcription of the albumin gene, and we would not have the appropriate activator combination present to facilitate transcription of the crystalline gene. And then if we were to look at an eye lens cell, we would see the inverse. The correct transcription factors for or activators for the crystalline gene, but not for the albumin gene. Okay, so in addition to that, there are a lot of mechanisms for protein modification and RNA modification after transcription. So we've already looked at one of those, which is the RNA processing step and alternative splicing. So that can determine the level of activation of whatever particular protein is produced. And then in addition to that, protein activation itself is another mechanism for regulation of the processes associated with gene expression. And that typically happens when a protein is either phosphorylated or dephosphorylated. And we did see an example of this, proteins getting activated through phosphorylation earlier on when we were talking about the process of signal transduction. Okay, so that takes you through some of the methods that eukaryotic organisms use to regulate gene expression, and hopefully just gives you the sense that there's really a broad range of ways that expression of any given gene could be regulated in a eukaryotic organism. So that again helps us to start to understand why it is that cellular function differs across different groups of cells within an individual, in space or through time, or between individuals. So we'll wrap up this section then considering gene expression over the course of development and some of the major patterns that people have observed with regards to gene expression patterns during development.